Welcome to your comprehensive interactive study breakdown of cell-level systems. This topic is the bedrock of GCSE biology, covering everything from the microscopic structure of cells to the vital processes of photosynthesis and respiration. By the end of this video, you'll be able to distinguish between eukaryotic and prokaryotic cells, explain how substances move across membranes, and describe how plants and animals obtain energy. Let's dive in. Let's map out the key cell types. All living organisms are made of cells, but they fall into two categories: eukaryotic cells, which have a nucleus and organelles, and prokaryotic cells, which are simpler and lack a nucleus. Animal and plant cells are eukaryotic, but plant cells have extra features: a cellulose cell wall for support, a large permanent vacuole for turgidity, and chloroplasts for photosynthesis. Bacteria are prokaryotic. They have a single circular DNA and sometimes plasmids, but no nucleus. Remember, examiners love to ask you to compare these cell types. Turning to our core study notes, substances move across cell membranes by three main methods. Diffusion is the passive movement of particles from an area of high concentration to low concentration, like oxygen entering cells. Osmosis is a special type of diffusion for water, moving from a dilute to a more concentrated solution through a partially permeable membrane. Active transport is the opposite. It moves substances from low to high concentration, which requires energy from respiration. This is how root hair cells absorb mineral ions from the soil. Be ready to explain these processes in exam questions. Now let's understand enzymes. They are proteins that catalyze chemical reactions in cells. Each enzyme has a specific shape, including an active site where the substrate binds. Temperature and pH affect enzyme activity. As temperature increases, rate increases until the optimum, then it drops sharply as the enzyme denatures. Denaturation is a permanent change in the active site shape, so the substrate can no longer bind. The same happens with extreme pH. This is a common exam topic, so make sure you can interpret graphs of enzyme activity. Let's work through a 6-mark exam question on photosynthesis. To get full marks, you need to explain that the rate of photosynthesis is limited by the factor in shortest supply. For example, on a graph, if light intensity increases but rate plateaus, then CO2 or temperature is the limiting factor. Always mention that enzymes involved in photosynthesis can be denatured by high temperatures. Practice interpreting these graphs. They are a favorite in exams. Watch out for common pitfalls. Many students mix up diffusion and active transport. Remember, active transport needs energy and moves substances against the concentration gradient. When describing osmosis, always mention partially permeable membrane. And for enzymes, make sure you say denaturation is permanent and changes the shape of the active site. These small details can cost you marks. Also, when comparing cell types, list specific organelles for each. To wrap up, remember the key differences between eukaryotic and prokaryotic cells. Understand the three types of cell transport and when each is used. Know that enzymes are sensitive to temperature and pH, and that denaturation is irreversible. For photosynthesis, always consider limiting factors. And finally, aerobic respiration is the process that releases energy in cells. Connect these concepts together, they all build on cell structure and function. You've got this.